Hello students, today we are going to learn about MSc molecules. First, we'll have a brief introduction. MSc molecules are membrane-bound glycoproteins encoded by the major histocompatibility complex MSC, which bind peptide fragments derived from pathogens and display them on the cell surface for recognition by appropriate T cells. The consequences are almost always deleterious to the pathogen. Virus-infected cells are killed, macrophages are activated to kill bacteria living in the intracellular vesicles, and B cells are activated to produce antibodies that eliminate or neutralize extracellular pathogens. The major histocompatibility complex is a collection of genes arrayed with a long continuous stretch of DNA on chromosome 6 in humans and chromosome 17 in mice. The MSC is referred to as the HLA complex in humans and H2 complex in mice. Although the arrangement of genes is somewhat different, in both cases the MSC genes are organized into regions encoding 3 classes of molecules. Now let us see the functions of MSC in immune response. They play roles in intercellular recognition and in discrimination between self and non-self. They participate in the development of both humoral and cell-mediated immune responses. Most T cells recognize antigen only when it is combined with a MSC molecule. They act as antigen-presenting structures, the particular set of MSC molecules expressed by an individual influences the repertoire of antigens to which the individual's Th and Tc cells can respond. It partly determines the response of an individual to antigens of infectious organisms, and it has therefore been implicated in the susceptibility to disease and in the development of autoimmunity. Natural killer cells express receptors for MSC class I antigens, and the fact that the receptor-MSC interaction may lead to inhibition or activation expands the known role of this gene family. The rich polymorphism of the MSC region represents a species response to maximize protection against diverse microorganisms. An apparent example is the malaria-driven selection at the HLA-B locus, whereby resistance to severe malaria resulting from strains of Plasmodium falciparum in East Africa is associated with HLA-DRB1 0101, whereas HLA-DRB1 1302 confers resistance to West African strains of the parasite. The MSC encodes 3 major classes of molecules. 1. Class I MSC genes encode glycoproteins expressed on the surface of nearly all nucleated cells. The major function of the class I gene products is presentation of the peptide antigens to Tc cells. It consists of a heavy polypeptide chain of 44 kilodalton non-covalently linked to a smaller 12 kilodalton polypeptide called beta-2 microglobulin. The largest part of the heavy chain is organized into 3 globular domains which protrude from the cell surface. A hydrophobic section anchors the molecule in the membrane, and a short hydrophilic sequence carries the C-terminus into the cytoplasm. Second one, class II MSC gene encodes glycoprotein expressed primarily on antigen-presenting cells, macrophages, dendritic cells, and B cells, where they present processed antigenic peptides to Th cells. They are transmembrane glycoproteins, in this case consisting of alpha and beta polypeptide chain of molecular weight 34 kilodalton and 29 kilodalton respectively. Structural studies have shown that the alpha-2 and beta-2 domains, the ones nearest to the cell membrane, assume the characteristic Ig fold, while the alpha-1 and beta-1 domains mimic the class 1 alpha 1 and alpha 2. The third one is the class 3 MHC genes encode various secreted proteins that have immune functions, including components of the complement system and molecules involved in inflammation. A notable cluster involved 4 genes coding for complement components, 2 of which are for the C4 isotypes, C4a and C4b, and the other 2 of the C2 and the factor B. The cytokines tumor necrosis factor TNF, sometimes referred to as the TNF-alpha, and lymphotoxin LT-alpha and LT-beta are encoded under the class 3 umbrella, as are 3 members of human 70 kilodalton heat shock proteins. Cellular distribution of MHC molecules. Class 1 MHC molecules are expressed on most nucleated cells, but the level of expression differs from different cell types. The highest level of class 1 molecules are expressed by lymphocytes, where they constitute approximately 1% of the total plasma membrane proteins, or some 5 into 10 to the power 5 molecules per cell. In contrast, fibroblast muscle cells, liver hepatocytes, and neural cells express very low levels of class 1 MHC molecules. MHC molecule can bind many different peptides since the MHC alleles are codominantly expressed. A heterozygous individual expresses on its cells the gene products encoded by both alleles at each MHC locus. The expression of so many class 1 MHC molecules allows each cell to display a large number of peptides in peptide binding clefts of its MHC molecules. Unlike class 1 MHC molecules, class 2 molecules are expressed constitutively only by special antigen presenting cells, primarily macrophages, dendritic cells, and B cells. Thymic epithelial cells and some other cell types can be induced to express class 2 molecules and to function as special antigen presenting cells under certain conditions of the understimulation of some cytokines. Peptide binding cleft. The classical MHC1 and 2 molecules contain a peptide binding site at the distal end of the molecule from the membrane. This peptide binding site is formed of 2 protein alpha helices lying on top of a beta pleated sheet. The binding site, or the groove, as it is often known, can accommodate a peptide of about 9 to 10 amino acids in length. Although for class 2 MHC molecules, the ends of the groove are open, allowing longer peptides to extend out of the either end. A wide variety of different peptides can be bound tightly by interaction with the conserved residues in the MHC molecules and the amino acid backbone of the antigen peptide. In order to accommodate the side chains of the larger amino acids, however, the floor of the groove contains a number of pockets. It is the size and the position of these pockets which limit the range of peptides which can be accommodated. This selectivity in peptide binding means that in each individual, only a small portion of each antigen can be presented to the T cells, focusing the immune response onto only a few defined epitopes. Now we will come to the MHC genes. H2. The MHC of the mouse carried on the chromosome 17 have roles in transplant rejection and normal cell interactions. KDL. The class 1 genes of the H2 coding for a chain molecular weight 44,000, which in combination with beta 2 microglobulin makes up the 4 domain K, D, and L molecules. Because any cell can potentially be infected by a virus, class 1 antigens are expressed on virtually all cells in the body with the exception of red blood cells in some species. AE, the classical class II genes of H2, which present processed peptide antigen to the antigen receptor of CD4 T cells. A and E contain separate genes for the alpha molecular weight 33,000 and P molecular weight 28,000 chains of the 4-domain molecule. HLA, the human MHC on chromosome 6, closely analogous to H2 except that the class I genes lie together and there are 3 class II genes. ABC, the classical human class I genes which present processed peptide antigens to the antigen receptor of the CD8 T cells. A is the homologue of K in the mouse. DP, DQ, DR, the classical human class II genes which present processed peptide antigens to the antigen receptor of the CD4 T cells. The distribution of these different isoforms within the body is slightly different, but it is still unclear whether each one plays a distinct role in the regulation of T cell responses. Class 1b genes, these include QA and TLA in the mouse and E, F, G, H, J and X in the human. They play a part in controlling the innate immunity, perhaps by regulating NK cell activation. The class II region, these include DM and DO, H2O and H2M in the mouse, class II MHC-like molecules which regulate the loading of the peptide fragments onto DP, DQ and DR. The region also contains LMP genes and the TAP genes. Beta-2M, beta-2 microglobulin molecular weight 12,000 coded quite separately from the MHC, nevertheless forms part of all class I molecules, stabilizing them on the cell surface. HLA-associated diseases. Many diseases show genetic associations with particular HLA alleles. Examples: number 1, rare sleep abnormality, narcolepsy, which virtually only occurs in people carrying the DR2 antigen. The reason is quite unknown. Arthropathies involving the sacroiliac joint, ankylosing spondylitis, Reiter's disease, etc., where one HLA B27 is found in up to 95% of the cases, nearly 20 times its frequency in the general population. Numerous other diseases, including almost all the autoimmune diseases, show a statistically significant association with particular HLA antigens or group of antigens, especially in the class II region. The explanation probably lies in the ability or otherwise of the HLA molecule to present particular microbial peptides or alternatively self-antigens. Finally, the conclusion. The major histocompatibility complex MHC comprises a stretch of tightly linked genes that encode proteins associated with intercellular recognition and antigen presentation to T lymphocytes. Class I MHC molecules consist of a large glycoprotein chain with 3 extracellular domain and a transmembrane segment, and beta-2 microglobulin, a protein with a single domain. Class II MHC molecules are composed of 2 non-covalently associated glycoproteins, the alpha and the beta chain, encoded by the separate MHC genes. Both class I and class II MHC molecules present antigen to the T cells. Class I molecules present processed endogenous antigen to CD8 T cells. Class I molecules are expressed on most nucleated cells Class 2 antigens are restricted to B cells, macrophages, and dendritic cells. The class 3 region of the MSC encodes molecules that include a diverse group of proteins that play no role in antigen presentation.